Hello and welcome to the program. Thanks for joining us. My name's Sam Ankerson. I'm the director of the Bailey Matthews National Shell Museum and Aquarium. And we're here for Steve Geiger's talk, What Can Oysters Tell Us About the Restoration of the Everglades? We're very glad to have Steve with us. He's a good friend of the museum and has been for a long time, a sometimes collaborator with our science director and curator, Jose Leal, and on various projects and studies. And he's an accomplished scientist who's been studying mollusks and marine systems and all kinds of things in our neck of the woods for a long time. And in recent years, he's had a special focus on oysters and bay scallops and general kind of gastropod abundance. And tonight, we'll be focusing on the important subject of oysters, that critical indicator species, the workhorse mollusks that they are, and connections to what's going on in the Everglades. This is we're about midway through our summer and fall Zoom lecture series. And we have our next program coming up is on Thursday, October 16th. The speaker is John Fallon, who is the Director of Sustainability and Conservation Initiatives at the Audubon Nature Institute in New Orleans. And the subject is Gulf seafood, sustaining wildlife, and our way of life. And that is, as this is, that's a free talk. You can go to the museum's website, shellmuseum.org, and sign up for that, just the way you signed up for this. Or if you're getting it through the emails, those will come to you as well. Also on our website, on the lectures section, is a video archive of all prior Zoom lectures dating back to, I believe, 2021, perhaps even 2020. And there's about 30 there now, and some on a wide range of subjects. And those are always there, free and available for you to access. I want to thank our supporters of this lecture series, and there are two of them. There is the Sam and Francis Bailey Clean Water Education Center here at the museum, which is a new initiative established this year to build awareness and advocacy for clean water, subject of clean water and clean water issues in partnership with Sanibel Captiva Conservation Foundation and Captains for Clean Water, the Sanibel Captiva Chamber of Commerce as well. And there's also support for educational programs this year from the Glenn W. Bailey Foundation. And now, well, actually, right before I introduce Steve, and if Steve can hear me, if he wouldn't mind turning on his camera and his mic, because I'm about to introduce you. But hey, Steve.
Hey,
Um, but questions and answers for Steve's talk, please ask away. And the way to do that in this Zoom webinar format is to use the chat function. So you would run your cursor along either the top or the bottom of your screen and click on chat and type in your question. And after Steve's presentation, we will get to the questions one by one, but please feel free to ask/type them in at any time. And so with that, we'll introduce Stephen Geiger, Ph.D., who's a research scientist, molluscan fisheries, at the and marine fisheries biology for the Florida Fish and Wildlife Research Institute at the Florida Fish and Wildlife Conservation Commission. He earned his Ph.D. in marine science from the University of South Florida, his MS in marine science also from the University of South Florida. And as I mentioned earlier, he has been doing a lot of good work for a long time, 30 years running at least. And he's a great scientist and advocate for mollusks and all around good person as well. So thank you, Steve, and we'll hand it over to you. And thank you all for joining the program.
Awesome. Thanks, Sam. That was really gracious introduction. I can't thank you enough for having me here. It's always great to share the work that we do, and of course, thank the museum and Jose for some of the collaborative work that we have done. It's hard to underscore how valuable resources like the Shell Museum are. So again, thanks for that. Just going to go ahead right into the next slide. Ideally, I'll advance. So just thought I would start with a little introduction. Sam gave you a little brief taste of that. My undergraduate work was done on Long Island, very different environment. I was a little school called Southampton College, part of Long Island University, and actually started working on oysters in the mid 1980s. It's a long time. There are oysters and scallop fisheries on Long Island. My master's work moved to Florida in the 80s and spent a lot of time in the Everglades, especially, or the edge of the Everglades, Rookery Bay National, I think it was an estuarine reserve at the time. That work was on tarpon, totally different work for my PhD work. I was fortunate enough to have a professor who was funded by the National Science Foundation to study the Antarctic and some of the ways that the ice edge and the Antarctic ecosystems function. So the fish that you see on the lower right-hand portion is called a myctophid, one of the most abundant organisms on Earth. You may have heard them called lanternfish. So got to just do some amazing things as a grad student. At the same time, I started working as an intern on tarpon and then later plankton, similar to some of the projects that study red tide today at the Fish and Wildlife Research Institute. It was part of Department of Environmental Protection and or Department of Natural Resources. So our titles changed a lot, but I have been in this building for quite a while. When I started officially as a full-time employee, which was 2001, some of the people I'm sure on the call today remember hard clams and calico scallops used to be a really abundant fishery. They were in a state of decline. Bay scallops were in an active restoration phase. So when I started, there was only a very limited recreational harvest of scallops, and part of our program helped to restore those scallops, a fishery that is still open recreationally today. And then over time, did a lot of work on gastropods, and I'd be happy to talk about some of that another time. So that's just a little background into my history. Essentially, my job at Florida Fish and Wildlife is to just have expertise in mollusks. Our focus lately has obviously been oysters. If you've followed the news in Florida, oysters have gotten a lot of attention. We do a little bit of work on scallop still. Again, off topic, just there's a lot to cover. There are a lot of mollusks in Florida. Today, I will focus on oysters. So I'm going to fumble a lot of this. Everglades restoration is really complex. I'm an oyster biologist, and I will spend a lot of time talking about Everglades restoration in general, because you have to have a little bit of background, maybe a lot of background, maybe too much for some of you, to understand why we study oysters and the value that they have. So first, I'll explain what is SERP. You may have heard it. Anything associated with Everglades restoration has a lot of acronyms. So I will explain what SERP is, give you a little bit of overview about our monitoring program, some of the major environmental issues that both affect oysters and maybe are influenced by oysters, and then try to talk a little bit about trends. And I will throw a lot of caveats as I go through my talk. Whenever I talk about our program, some of the people who are familiar with either Bailey Matthews or Sanibel Captiva Conservation Foundation will remember the name Aswani Valetti. He had a lot of different titles at Florida Gulf Coast University, but he essentially helped us get introduced to monitoring oysters in the Everglades. And then I also like to give a shout out to Melanie Parker. A lot of the data that I talk about, Melanie Parker used to work for us at Florida Fish and Wildlife. She now works at the Water Management District. She's really responsible for the design and just the excellent organization that we still rely on. So I try to acknowledge those two people, really instrumental to getting this program started. So Comprehensive Everglades Restoration Plan, the acronym is called SERP, and it is essentially Everglades restoration was built and developed as probably a 50-year project. Anybody that knows the history of South Florida knows we have changed the way that water flows. I'm going to try the laser pointer and see if this works. And historically, most of you have seen water flowed south from Lake Okeechobee through the Everglades and what is called sheet flow. That may or may not be exactly precise. People are still studying that, but over time, what happened was the channels were developed to move water away from South Florida. There were two reasons for that. One was to prevent flooding, and one was to allow more agriculture and development. So more water flows through the Caloosahatchee River and through the St. Lucie River, amongst some other minor flows. And the plan for Everglades restoration is to try to return some of that flow to a more natural system. So that is the goal of this. There are lots and lots of documents. If you ever want to look for it, evergladesrestoration.gov. I think that website is sponsored by the Army Corps, but more information than I can comprehend, and lots and lots available. The Army Corps, the Water Management District, and all of the parties involved get together to periodically give an overview to Congress. So a lot of this is funded jointly between the state of Florida and Congress, US government, primarily through the Army Corps. And we are required, we being the community, to update Congress on the progress. And we just put out a system status report, essentially, how are things going? What is happening in the Everglades? Are we meeting our goals or not? So if you just Google Everglades System Status Report, you can find that, and the links here that will be in the talk. I wanted to put this up here just for people that are not familiar with Everglades restoration, how complex the system is. I cannot personally keep track of all the projects that are ongoing, so the map on the left just gives you some idea. It's really overwhelming to try to understand everything that is going on. So if I fumble and I don't do a great job, you're more than welcome to call me and say, hey, you didn't cover this aspect that well, or you misspoke. It's really, really a lot to comprehend. So I wanted to show two of the projects that you may have heard about. The C-43 storage is as a reservoir or a basin, which is intended to store water when there's a lot of water flowing. This was officially opened this spring or summer. And then another project that you may or may not be familiar with in the lower box on the right is the Picayune Strand. This is an area where they're trying to restore a little bit of flow locally. And then anybody has ever driven over either US 41 or Alligator Alley has seen some of the bridges. So all of those projects are some of the smaller projects. C-43 is pretty big, but intended to restore flow to the Everglades. So things are starting to happen, which is cool. Everglades restoration is monitored under a program called Recover. And this is basically a plan for how we monitor projects. This, you can see in the box on the right, there are a lot of ways in which the progress of Everglades restoration is measured. By my count, there are 48 ecological indicators, some of which have 10 or more metrics. And I'll explain what our metrics are in an upcoming slide. So it's a lot. There are a huge number of people involved in this project. There are 4 major regions for which the monitoring is grouped. They're called the Northern Estuaries, which is essentially Loxahatchee, St. Lucie, and the Caloosahatchee River. The Lake Okeechobee District, the Greater Everglades District, and the Southern Coastal Systems. And there's another project coming up for the Southwest Coast. So just a ton of stuff to cover. If you focus on just our box, the Northern Estuaries, this is where we work, the St. Lucie, the Loxahatchee, and the Caloosahatchee River. One of the things that I wanted to point out is that when you're when you're planning how to manage Everglades restoration, there are a lot of competing interests just among the natural environment, not counting the stakeholders involved. Um, Oysters and the seagrasses don't necessarily have the same ideal conditions. So you can do things that help oysters that may not be great for seagrass. The same can be said for the many species of seagrass. Widgeon grass and turtle grass may or may not have the same requirements for the environment and the species that are associated with them. And then when you add in the upland areas, uplands may have very, very different conditions, which are ideal relative to the estuary. So think about things like cypress domes, the little birds that nest on shorelines, the wading birds in the Everglades. They all have different seasons and requirements under which they do better. So it's just a very complex system to manage. Okay, so in the systems that I work in, I study oysters in what are called the northern estuaries. And you can see for the Caloosahatchee, we have aquatic vegetation, oysters. Chlorophyll and salinity are essentially measures of how the water quality is doing. In the St. Lucie River, we have the same metrics in the Loxahatchee. The main exception is that in the St. Lucie River, they also study benthic fauna. So things like worms and clams and really tiny fauna that live down in the mud. But I'm going to focus on oysters, obviously, for today's talk. The places that I work, St. Lucie Estuary, we have 9 stations. Loxahatchee River, we have 6. And then the Caloosahatchee River, which probably most of the people in this talk are more interested in, we have the 4 stations upstream and downstream of Shell Point. And that'll be important later. I'll try to talk through that as clear as I can. So some people, when they hear me talk, I have been called essentially Pollyanna because, you know, there are places that we monitor that are absolutely doing great. And depending on where you study, there are oysters that are doing really, really well. Other people have called me Chicken Little because there are places that we study that are not doing well. The reality is somewhere in between those. Some of the oysters in the ecosystem are doing great, and some are not. And it's just really complicated to talk about the overall system status in a single report, even if I'm just talking about oysters. So the principal metric that we use to monitor how well oysters are doing is the acres of oyster. And what I show here is just, it's a printing of a Google map that I have a layer for oysters. Now, we don't really study down in Estero Bay, but it is being affected by some of the restoration. You can see in the Caloosahatchee River, there are not a huge amount of mapped oysters. That doesn't mean there's not oysters there, but mapping is a really complicated task. One of the ways which the Water Management District and the Army Corps study the distribution of oysters is to pay for very extensive studies. And so in the panel on the left, well, I'll back that up. There are 3 studies which attempted to measure how many oysters were in the systems that I talked about, Caloosahatchee, Lake Worth, or Loxahatchee and St. Lucie. This is a map showing what has been measured in the Caloosahatchee and San Carlos Bay area. The panel on the left shows the difference between the 2003 and 2010 surveys. Anything that was in green, there were more oysters or better habitat, and anything that was in red, there were less. And then the panel on the right shows the change from 2010 to 2019. The problem is that's a long time in between surveys, and you don't necessarily have the same people doing those studies. So you have to be a little bit cautious about assuming that the metrics were measured in exactly the same way. And that's one of the problems with these long-term databases. Measuring change over long periods of time is very hard. So we were brought in to try to come up with a way to measure changes on a shorter time scale. We do a lot of surveys. In this picture, you see we do quadrat surveys, essentially go out and measure how many oysters are present on some of the oyster reefs. We measure reproductive status. I'll talk about that. We measure disease. I will not talk too much about disease. Settlement, which is comparable to reproduction, and then growth. I won't talk a lot about growth either because growth is kind of complicated. So when we go out and we do a survey, this is just a picture of our settlement strings. We call them T-bars. If you've ever seen those out in the estuary, they look a little different. In the Caloosahatchee River, we essentially only use one stringer. But what we do is twice a year, we go out and we measure a fixed sample station. So the same reefs we have been measuring since 2005. We basically count how many oysters are present, and then we measure a whole bunch of metrics on those. This is our survey. So this is just a couple of pictures of what the survey looks like. It's a huge amount of work. We have measured literally millions of oysters in the time that we have done this project. Every month we go to each of our stations and we bring back oysters for a variety of metrics, largely related to their health and their reproductive status. Part of that assessment is how many diseases and how many parasites they have. So it's just an example. From Caloosahatchee, we collect 5 oysters each month from each of our 4 stations. The picture on the left is an oyster that looks relatively healthy, a little bit watery maybe. The picture on the right shows the shells of an oyster where the tissue has been removed, but you can see things like mud blisters, which is a worm that affects the quality of the shell, and then a yellowing, which is a disease which detracts a little bit from the energy of the oyster. So these are things that we are measuring every month on lots and lots of oysters. Reproductive status is really important because if your organisms, whatever organisms you're studying, aren't reproducing, you will not have another generation. So on the top, on the left, what you see is a very ripe female. On the bottom, on the left, is a very ripe male. Oysters are what are called broadcast spawners. Because they're sessile, they don't move. They have to find a way to reproduce. So what they do is the females and the males release their gametes into the water column, and hopefully you have fertilization. So the diagram just kind of shows the gametes are released into the water. At really low densities, the sperm can't find the eggs. And so you have what's called, it's a density dependent decline. So when the density gets really low, you have low fertilization success. On an oyster reef, that is not generally a problem. If you've ever looked at the seawall or been out to an oyster reef, generally there's lots and lots of gametes. So fertilization success is pretty successful. Once the egg is fertilized, it takes about a day to develop into a swimming larvae, and then it spends somewhere, depending on the temperature and the food, between 10 and 20 days in the water column. The larvae are much more sensitive to environmental conditions. We don't do a lot of work with larvae just because it's so difficult and it's a really rapid process. What we study is settlement. So if you look at the very end of this picture, I'll do the laser pointer again. That settlement, when the oyster goes from the water column, from a swimming veliger down to the bottom, is a really important cue. They have to find substrate to settle on. Oysters are not as picky as some species. They will settle on just about anything, but their preferred substrate is actually other oysters. So when the oyster reef declines or is covered with mud or other fouling, the oyster larvae don't have as many places to settle. Every month, our combined total is something like 900 shells, and we count how many larvae are settling. In the history of this project, we've measured about 250,000 of these shells. This is just a picture of what the setup looks like. This is Celia holding up a harvested set of stringers. And then the picture on the lower right shows what is a good settlement, something like 50 to 60 spat that have settled on an oyster shell. Anywhere from somewhere around a millimeter, it's about as small as you can see with the naked eye, up to, in a month, they can reach almost the size of a quarter. So oysters grow really fast when the conditions are good. This is an important indicator. If we don't see spat, we know that there was an interruption in the larval phase where even if the oysters were spawning, if they weren't setting, something in the water column prohibited them. Either there wasn't enough food, there was too much predation, any number of factors. The next metric, I won't talk a lot about this, but what we do is we put oysters in these cages and we try to assess how fast do they grow each month and how many are predated. This is kind of a fun aspect of the project. This is just a sampling of the kinds of things we find in the shell. We find tropical fish, especially over in Loxahatchee. There's a sheep's head. Sheep's head are a big predator of oysters. You find little juvenile snappers. We have found lionfish. The middle panel on the left is drills and their eggs. Drills absolutely love clean shells to lay their eggs on, and they are a major predator of especially small oysters. The picture in the middle on the right, sometimes we come back and there's just nothing. We have no idea, were they consumed? Were they washed away by waves? We don't really know. The bottom left, that's a fishing jig. We find a lot of people must use the buoys to fish. I know sometimes there's triple tails or snook hanging around. The picture in the middle on the bottom is a stone crab, lots of crabs in our traps. And then the bottom right is just algae. There are times where we show up and our cages are just completely covered with algae. The main point that I want to make about this is even just the cages full of shell become habitat. Oyster is a really, really rich habitat, and I can't talk about the value as habitat too much. So I'm almost there. I'm also ready to actually talk about oysters. This is just a picture of what our setup looks like. So if you ever see this, these T-bars and our cages, growth cages, that's typically what it looks like. So you will see those around our estuaries. Before I actually talk about oysters, I have to talk a little bit about what the requirements are for oysters to survive. So these are called polygons for survival. This one on the top left is a salinity polygon for larval oysters. And what you can see is that the value of a habitat when the salinity is down around 6 goes to 0. And then as the salinity rises to about 15, you reach an optimal salinity for oysters, and then it goes higher. It's less optimal. The main reason for larval oysters is that as the salinity goes higher, they are predated. For adult oysters, the polygon is pretty much the same shape. And then for temperature, these may or may not be perfect, but below about 15, the larvae are not going to grow fast enough to survive to settle. For adults, they can certainly survive down to about 10 degrees centigrade. It really takes something like a frost to kill the oysters in Florida. They won't do anything, they won't grow, but somewhere around 10 degrees is what they need. And then I'll talk a little bit about temperatures as we go on. Once you get up to about 35 degrees or 100 degrees Fahrenheit, oysters are definitely not happy. They are stressed. In our estuaries, this top figure that you see here is just an example from the St. Lucie River from 2005 through 2024. Anybody that lives in Florida knows in the summer it gets wet and in the winter it gets dry. So this just kind of showing you this pattern over the years. Not every year is the same, but if you look at that data, the percentage of days where the salinity is lower than is optimal for oysters in the St. Lucie River is very variable. And a lot of this comes from things like 2005. You remember 2004 is not on this chart, but 04, 05 was a really bad hurricane year. So a lot of these years where the water was really low are associated with tropical activity, sometimes with stalled fronts or just wet seasonal weather. Here's just a shorter close-up view of this. One of the reasons in our estuaries, what you see right here is that when they are releasing lots of water from Lake Okeechobee through either one of the two major canals, that's when you drive the salinity in the estuaries, depending on where you measure, down to 0. And that's bad for almost everything that lives in the estuarine portion of the estuary. So a lot of the times you see these zeros, that's when you start having mortality of oysters, you start having some of the organisms pushed out of the river. Not ideal, but they happen. And there's a variety of reasons they happen. I'm happy to talk about that later. One of the caveats to that is that depending on where you measure in the river, the daily mean salinity is not necessarily the only thing that's happening. So in this graph, what I show is at a place called Speedy Point, which is a constriction in the St. Lucie estuary, that is exposed to tidal flow. So if you measure the salinity on the tide, on any given day, the salinity may vary by 10 or 15 parts per thousand. This is the ideal conditions for oysters. Oysters actually love a little bit of variance. If you have this kind of variance, that means there's flow. And so generally that is a condition that is going to favor oysters. Going back to the map of Caloosahatchee, what you can see are the 4 stars. We measure oysters upstream and downstream of Shell Point. There's some reasons for that, partly because the oysters at Shell Point are not easily accessible and they're not necessarily the greatest reefs. The conditions upstream and downstream of Shell Point are very, very different. And I'm going to show you some graph. I will put in a little plug here. We work with Sanibel Captiva Conservation Foundation. I use their data a lot. I try to put a plug in for Eric Milbrandt. He is the main person I work with over there. They run a data monitoring site called the Recon. If you Google SCCF Recon, you can find this data. Depending on where you are in the estuary, the salinity conditions are very variable. So this is a picture from their data collection from just a couple of weeks ago. The blue line is Tarpon Bay. If you know where the Marine Lab is, this is the mouth of Tarpon Bay. The salinity is almost always up around 30, so close to Gulf Ocean salinities. If you go upstream at the US 41 bridge on the Fort Myers side, right now, or when this data was collected, the salinity was almost exclusively down below 10. And you can see a little bit of tidal signature, but most of that is gone. If you look right at that constriction, the Shell Point salinity shows that optimal condition for oysters, where the salinity is varying. This might be a little more than they like, but that means that every day there is some condition which is probably pretty good for oysters. So again, where you are in the estuary will matter. I'm going to get a little bit nerdy here, and I need to point out that any of the oyster data I am presenting is not published data yet. This is a very long-term study, and so essentially what I'm presenting here are trends or patterns that I see. Eventually, we will go through the peer-reviewed process where we write up our results, we present it to other people in-house, and then other people out in the general public. Not general public, but the journals that we publish in will provide typically anonymous reviews. It's a really important process when you're publishing science to have those peer reviews where other people examine what you're saying and either agree or disagree, criticize. Sometimes you modify your analysis. The data that I'm presenting here are not peer reviewed yet. I just want to caveat that. In the graph on the top, what you see are, if you remember going back to that polygon, in the Caloosahatchee River, since about 2016, the salinity spends about 40% of its time in any given year below the optimal conditions. But if you look downstream, for example, Bird Island, San Carlos Bay, there are very few times where the salinity is below optimal. So again, depending on your perspective, some of the parts of the estuary are good for oysters and some not as great. The flip side of that is that the downstream sites can actually be too salty. So in particular in Tarpon Bay, it is rarely the case where the salinity is optimal for oysters. Now, high salinity is not quite the same for oysters as low salinity. The main issue with high salinity is that the predators and the disease have more time to affect the oysters. It doesn't kill them instantly, but it can, in the long run, degrade the quality of the reef. So even though the upstream conditions may be, for the last 10 years, a little more fresh water than is optimal for the upstream reefs, it may be beneficial for the downstream reefs. So again, when you're talking about the entire Everglades system, even amongst oysters, deciding the conditions that are going to exist in the estuary within the ability of the Army Corps to control is a difficult choice. It is just complicated. You'll notice 2024 here, everybody's data collection was impacted in 2024 because, as a lot of you know, we had a rough year weather-wise. Temperature is another thing that really affects oysters. This is just an example. It could be from any one of our estuaries. I'm pretty sure this one is actually St. Lucie. It's cold in the winter and warmer in the summer. The reason that I picked 20 degrees and 30 degrees, when the temperature goes below 20 degrees centigrade, that's when oysters essentially stop reproducing. When it goes above 30 degrees, that's when oysters start to be stressed by hot temperatures. I'm sorry, in this graph, if you just look at that graph, it's really, really hard to look at the graph and say, well, looks like conditions have kind of stayed the same. It doesn't seem to be changing. But when you dig into the data a little bit more, what we are seeing, and this is data from Caloosahatchee, is that since we started studying oysters in the Caloosahatchee estuary, there are more days where the temperature exceeds 31 degrees. So um, I, I'm sorry, I think this was 2023 was the last of the collection data here. What you see is that in 2023, something like 25% of the days in that year exceeded 31 degrees centigrade or 88 degrees Fahrenheit. That's a big increase from what it used to be. On the bottom graph in blue, what you see is that the number of days where the temperature is below 20 degrees is falling. So what is happening is they're spending more of the time, let me rephrase that. The winters are staying warmer. That does not allow the oysters to rest. Oysters need that winter rest when they're not reproducing to be prepared for the reproductive season. So when it gets warmer and warmer, they don't have that recovery period that they need. And then, of course, in the summer, when you start having temperatures above 31, 32, it is stressful both for the adults and it reduces the survival of the larvae. So the question that people typically ask is, are the number of oysters changing? So when I talk about the number of oysters, I talk about the density. So when we do our quadrant, which is a quarter square meter, are there less oysters or not? I'm going to waffle again, and I'm going to make this complicated. I apologize. You're welcome to throw questions at me at the end. In the St. Lucie estuary, the upstream and downstream have very different conditions. So in the top graph in St. Lucie, the mid portion of the estuary, where you get that tidal signal, it's variable, but in general, the middle portion of the St. Lucie estuary is doing pretty good. The same for the Loxahatchee. It's kind of hard to pull out a pattern. In the upstream sites in the St. Lucie, it is generally not ideal for oysters. We did that on purpose because if they change the way that water flows, we should see recovery in the upstream portion of the St. Lucie sooner. If it doesn't change, then we won't. The middle estuary is probably going to be okay for a long time. Where you would expect to see the improvement is the upstream. So we have a long-term database to say, is it improving or not? In the Loxahatchee River, what you see is that the downstream sites actually are probably declining, especially during the last 10 or 12 years. The Loxahatchee River is not getting enough fresh water in the downstream sites. And so it's a little bit too salty. It's tough to say in the middle estuary if it is or not, but you could certainly argue that over the last 5 or 6 years, the entire estuary has declined. In the Caloosahatchee estuary, what we see is when we took over the program from Florida Gulf Coast University, there was a shift, and it's something I try to be really cautious about. If you just look at the upstream sites, it seems like over the course of this study from 2005, where there was nothing present, that was a really bad water year. So essentially, if you look at 2006 through 2009, through today, the upstream sites almost certainly have declined in quality, in the number of oysters that are present on those reefs. It seems like the downstream sites have too. And there's a lot of reasons for this. It's not necessarily related to Everglades restoration or the slow progress. Here's a couple of things that may be affecting the reefs. When the salinity is too high, the picture on the left are oyster drills. We see this especially in the Lake Worth Lagoon, in the Loxahatchee, and St. Lucie. Our East Coast sites in general have more predation than it used to from these oyster drills. We're not 100% sure why that is, but it's almost certainly the density of the predators have changed. Another thing that we see more of lately, especially at the higher salinity sites, is this orange sponge called Cleona. Cleona is really important because it is actually called a boring sponge. So it drills holes into the oyster shell and degrades the quality of the shell, which affects the longevity of the shell, which affects the quality of the reef. So these are things which are negatively affecting oysters, um, especially in the Southwest coast. I know anyone that lives on the Southwest coast knows Southwest coast has been impacted by a lot of tropical storms over the last few years. This is an island that we call Bird Island. It used to be a beautiful little mangrove island, and this is what was left after the combinations of Irma and Ian. And I apologize for not remembering all the names, but this site has just been hammered. The flip side of this, which we don't really know for sure, is that when the sand was moved away, there's a lot more shell left exposed for the oysters to settle on. So the jury is still out that even though it affected the mangroves really, really badly, the oyster reef seems to be doing pretty well. Again, we don't really know in the long term. That's kind of why we're doing the studies. This has nothing to do with Everglades restoration, but you have to keep that in mind that other factors are affecting our oyster reefs. A final thing I'm going to talk about for now, this is a Google Earth image of the watersheds. If you know where the Southwest Florida airport is, if you look at the two images, you can see that the local watershed has also changed a lot. So as you urbanize some of these estuaries, the quality of the water and the rate at which the water arrives in the estuaries, and even to some degree, which estuary the water ends up in, is affected by these develop, uh, by development in Florida. So again, I don't really want to get into that. I'm happy to talk about it in the question session, but suffice it to say that Florida is changing. Another thing that we measure, which is really important, is the size of oysters. Again, this graph is from our East Coast samples. What we have seen from the start of our study, 2005 through 2008, 2009 through 12, 13 through 17, and then finally 18 through 22, is that we are losing some of our largest oysters. And so the conversations that you hear about, there are fewer oysters in the fished oyster populations, are also happening in some of the estuaries where there is no legal oyster harvest. So there are fewer and fewer legal harvestable oysters, even though there's no fishery. So this suggests that part of the problem we may be having with the oyster fishery is not just related to the fishery. It's not conclusive, it's not definitive, but we see this problem in all of the estuaries we study where there are fewer legal oysters. And I can tell you from watching the logs, our law enforcement officers do not report a lot of poaching from any of the Southwest or East Coast estuaries. There's some, of course, there are people taking oysters illegally, but it's not a huge factor. And so something else is happening with the larger oysters, whether they're not living as long or what, we don't 100% know, but that is a conundrum, something we're desperately trying to figure out. Oddly enough, if you look at the Caloosahatchee oysters, even though the oysters upstream are less abundant than they used to be, when they are present, we seem to have legal oysters. So one of the possibilities is that the conditions, when they are right for the low salinity sites, are better for growth. The oysters grow faster and they have uh, better condition when conditions are right. Even though they're dying, they return pretty fast, which is really encouraging and hopeful that if we can get the salinity right, maybe we can fix that. You see to some degree the same thing happening in St. Lucie estuary. So this is really encouraging. If you look at the green, that's an upstream site. Legal oysters is anything over 74 millimeters, which is 3 inches. When conditions are good, those legal oysters show back up. So really cool. I know this is a really busy graph and I apologize for that. There's just no way to show this very easily. This is a project I'm working with some of my coworkers, in particular, Erica Levine. Don't try to really read the axes on this. I'll try to show from 2007 at the top through 2023. along with what I talked about for temperature, what you are seeing is the red graphs show essentially the percentage of oysters that are reproductively active. So what you see is that when we started this project, there were very, very few oysters reproductively active for most of the year. But as you go on, a higher and higher percentage of oysters are reproductively active for more and more of the year. We don't really know what that means for oysters because it's really, really difficult uh, to be reproducing all the time. It takes a lot of energy to reproduce. So part of the issue that may be happening with smaller oysters is that they're spending more time reproducing and less time growing. That's not definitive, that's not conclusive, but the evidence suggests that that is a possibility. Um, Again, it's something that is happening. We don't know for sure, but we have a lot of data and we're trying to figure this out. To go back to the maps that the long-term occasional surveys do, we are trying to institute what's called a randomized design. So the principal thing that I've been talking about all through this are fixed station designs, and it's a statistical quirk where if you want to look at a trend, it's best to monitor the same thing over and over and over again, the same way through multiple years. If you really want to understand what's going in a population, randomized designs give you a more accurate picture of the entire ecosystem. So we are trying to transition to part of our study is what's called a randomized design. We are doing this in the Caloosahatchee River as well. Suffice it to say, in the St. Lucie estuary, when we did this in 2023, only about 10 of the 33 historic locations had live oysters. We surveyed something like a quarter of all the known oyster reefs. So a little bit disconcerting, but it was a one-time study, just trying to work out protocols to try to figure out how to do this better more often. Okay, I'm sorry if I threw a lot of data at you and I went way too fast, or if I talked way too long, but I'll just try to summarize this. The major changes in the water storage and flow are starting to happen, and Everglades restoration is really reaching a point where hopefully we start to see some changes within the systems. But keep in mind that restoration of the Everglades is only one factor that will affect how the estuaries function. There are lots of other things going on. There are changes in the management and, in essence, how the water is released and the water flow that have been occurring since 2017. That's called the release schedule. So when they release water, how they release water, etc. One of the things that's really encouraging to me is that there is now an oyster biologist in the water management district, and she communicates that oyster biology to the teams. So oyster biology is being considered whenever they talk about how much water is released. The northern estuaries appear to be warming. The reproductive season seems to be prolonged. Again, I'm hoping to finish up that analysis over the next year or two. Not exactly sure how that affects the population. Is that good or bad to have more reproduction spread out over a longer time? I can't give you an answer on that. I'm hoping to find that too. The main thing to take home from this is that we have a really solid baseline, and major changes ideally are about to start happening for these estuaries. With that, I want to give a special thanks. A lot of the pictures that you saw were taken by Chris Kirby. He is the person that leads a lot of our field work and does a lot of the sample collection. Erica Levine has helped me with a lot of the data management and the reports. And then my current team, Lily, Darian, Olivia, Celia, Reina, and Nicole are the ones who really actually get most of the work done. So I can't thank them enough. Funding is provided by the Water Management District and by Palm Beach County. And with that, I'd be happy to try to answer any questions.
Thank you, Steve. All right. Great presentation. There are a couple questions I've seen in the chat, but one question I have before we get to those, and would encourage anyone with questions to type them into chat, and we'll get to them. Is for a lay person, are there some ways you'd recommend that if people wanted to sort of keep up on what's happening with with this science and kind of what's going on? I mean, you mentioned SCCF as, their Sanibel Captiva Conservation Foundation, um, their monitoring is a resource. That's one, but are there other ways that people can stay connected to it?
Yeah, I mean, the system status report has a huge amount of information. It's a little bit much to digest if it's the first time you've ever done this. It does have a summary page. It's a really long document. We try to do, especially things like I'm doing now. I try to get my message across. There are lots and lots of people that are working on Everglades. Do I have a great way to really keep in touch with that? It's hard. It is overwhelming just how many
Right.
moving parts there are to Everglades restoration. Um,
Right. There's not a one-stop shop for it,
I mean,
right?
unfortunately, the system status report only comes out. It's either 2 or 3 year intervals. Um, I mean, like I said, that's a lot of information to
Mm.
digest. No, I don't have a great one-stop shop. I'll talk to some of my partners who are much more uh, in tune with the entire Everglades restoration project. And
Mm.
I'm on committees, but man, it's a lot to keep up with.
Sure.
Um, one of I wanted to go back and touch on the work that SCCF does. Um, I'm not sure if people realize from what we're talking about. I talked a lot about restoration. We as an agency, I shouldn't say that. My division, which is the Fish and Wildlife Research Institute, we don't do a lot of the restoration ourselves. We essentially are tasked with monitoring progress of projects or the fisheries. We
Right.
have another division called Habitat and Species Conservation, which does actual restoration. We have a division called Marine Fisheries Management, which does restoration, but it's really enhancement of the fisheries. So for oysters, there are two very, very different goals. One is to create habitat, one is to manage the fished fisheries.
Mm.
Sanibel Captiva Conservation Foundation has a kind of a different role. They can advocate for change, um, but they monitor, but they can also conduct the restoration, and they have conducted quite a few oyster restoration
Mhm.
projects within the estuary. And so I try to draw that distinction where we have to work with partners because they can wear hats that we can't necessarily through legislation. And so they play a
Mhm.
really important role that just the way the laws are created, we can't necessarily do.
Mhm.
So, I mean, I can't thank Eric enough for some of the work that he's done. Um, Again, that it's a little bit different perspective, but
Yes.
uh,
Yes. No, understood. Yeah. No,
that might not
SCCF.
answer your question, but
Yeah, you did, and would concur. SCCF does phenomenal work across the board. All right, a couple questions here. All right, we can start with a question from Jose. Do you have any data or comments on the effects of the storm surge and Hurricane Ian in general on oysters at the Caloosahatchee Estuary and San Carlos Bay?
yeah. So I don't have uh, those what we call acute those instantaneous events. Essentially, hurricane for us is really a moment in time, and so salinity wise. Um, Something like a storm surge it does not hurt oysters at all from salinity point of view.
Mm.
They love they for the most part, with the exception of disease and predation, but they can handle the salt water. The hurricanes that have hit have not generally caused the kinds of water releases that really are detrimental to oysters. They can if they occur on top of an already wet season. So if you're having a very wet year and the
Mm.
lake is already full, and then you have a huge amount of rainfall. So for example, Helene and Milton dropped huge amounts of rainfall on parts of central Florida. And so when the ground is already saturated, the hurricanes absolutely can, but they don't necessarily. And so every storm is a little bit different. What
Mm.
we did see, and the upstream stations, the oyster reefs themselves had very little impact after the storm, in my opinion. There just wasn't a huge impact. the two sites in San Carlos Bay, um, one of the sites, which is really close to the Punta Rasa Bridge, the reef actually, to some degree, looked better. And what happened was a lot of the mangrove root oysters broke, and they are
Mm,
now on the bottom. And so the measures that we measure, that oyster reef seems to have done okay. And then I think I mentioned, and I'm sorry if I forget which storm was which, but one of the two storms scoured a lot of the sand off the Bird Island reef. And so that sand went somewhere, but for our reef, that exposed shell means there's more sediment substrate.
mm,
And so interpreting the effects of something like a short-term event like a hurricane on the oyster reefs is a little bit tricky. Um, if you're studying mangroves, it's different. Mangroves were decimated. I mean, mangroves got hit really hard, and mangroves continue to die for some period after the storm because they can't handle submersion. So when you get this
mm,
really high storm surges, my understanding from the people that study mangroves is that that causes harm to the mangroves that persists for months, if not years, after the event.
mm.
And also they get the soil washed out from under them. So depending on what you're studying, can have really different effects. For oysters, I can't say that the hurricanes, specifically just the event itself, were catastrophic, for example.
Right,
It doesn't mean there's not some other oyster reef that we studied that didn't get buried,
right.
because that sand went somewhere, right? So
Yeah, yeah,
um,
right.
that's
They're
a,
all right. They're
yeah,
all different,
yep.
right? All these events. Alan has a question. Are introduced oyster species a problem or an opportunity in Florida?
Yeah, so there is some question about the presence or absence. It's not an introduced species. There are many species of oysters, but there's one called Ostrea equestris, the crested oyster. Really, really difficult, even for me, to tell the difference between the two. Morphologically, they look really similar, but it is technically a different genus. Um, my feeling is that those will start to show up in the estuaries that are saltier. That is not an introduced species. So far, we don't have problems with introduced species of oysters in Florida. They tried to introduce a species for commercial in the Chesapeake. It did not do well. It's called Chrysostrea geiger, a Pacific or Ariakensis, sorry. It's a species from another part of the world that is aquacultured in other places, did not do well in the Chesapeake, and it will probably not survive in the temperatures that we have in the Gulf of Florida. Um, there are a couple of offshore species. Again, one is called Hyatissa. Probably not going to be a problem in the estuaries, but it does show up in offshore reefs, and just there's no baseline to even understand if that particular introduced species is out competing or filling a niche. For example, like is it out competing spondylus, for example? They're
Mm,
just not great data when you get into the offshore environments. It's just so hard to study, a deeper reef. So
mm,
far, no. I mean, knock wood. There are some parasites that we worry about um, present in oysters. Um, Perkinsus is one of the diseases I didn't really talk about. Perkinsus may have come from someplace else, but it has been in the US for about a century. And then, especially as you go farther north, there are a couple of other species which really haven't been a huge issue. Perkinsus or dermo absolutely is a problem for oysters. It is always present in the Gulf of Mexico. Essentially, if there are oysters present, they have the disease in the population, not necessarily every individual. Um, and so, so far that is a bigger problem. Other species, like I talked about, like the Cleona sponge, which may be introduced, I'm not 100% certain. And then some of the crabs and snails are probably a bigger risk than the oysters being introduced themselves.
mm,
Another thing that I'm sure people are familiar with is green mussels. Green mussels don't really seem to outcompete oysters on the oyster reef. They seem to do really well, like, for example, on the bottom of things that float, like boats and floating docks. Those are not ideal places for oysters, although they will grow there. So even though green mussels have shown up and they're persistent, I don't think they're really out competing. So, there are other species that potentially could. Um,
Mhm.
I mean, I hope that answers the questions. There are other
Yeah,
invasive species that are probably worse.
yeah, gotcha. Kellen has a few questions. The first is, which mollusks present, in your experience, are most indicative of good water conditions?
Oh, I mean, yeah, that's a tough.
I mean, oysters kind of have that reputation, of course, right?
Oysters definitely have that reputation, and they are not the most sensitive. I'm going to take a weaselly answer on that. In Florida, we have something like 300 species of bivalves, maybe more. And then depending on how you count it and where you count state and federal waters, somewhere around 1,400 species of gastropod. What I would say to that answer is that in most of our systems, the higher the biodiversity, in other words, the number of different species that are present, in general, is probably the healthier habitat. Now, of course, coral reefs are going to be more diverse um, than an estuary, especially the farther north you go. But um, I would probably not pick any one species. I would say a good indicator, which is very hard to study, is how diverse the community is.
Hmm.
Um, and if you've ever, for example, looked through your own website, there are a lot of itty bitty tiny mollusks
Hmm.
that we know virtually nothing about their biology.
Yeah, right,
Um,
right.
it's just um, the number of gastropods, as an example, that are funded for study in Florida um, right now are queen conch, and I have a little project I work with Jose on a little species of cone, and that's a very tiny grant. Um,
Mm,
so gastropods in general have not much money. There are not many other bivalves in Florida besides oysters and scallops that are
mm,
funded. So when you think about all those species, there really isn't much funding to even do this. So
mm.
biodiversity, I would say, is probably a little bit different answer maybe than you were expecting, but that's uh, probably where I would go to answer that question.
Yep. All right. I'm going to trust that you know maybe what this means, and I don't. Do you employ any VR technology for oyster monitoring? Is that virtual reality? Was that what VR would be?
I'm going to assume uh, it depends how you define that. We certainly use things like data loggers. The recon system is an example of a data logger. So you leave a piece of equipment in the estuary. We do use things that measure temperature, salinity. Um, I can't remember if SCCF does it, but here at FWI we have a system um, which measures plankton, and so they're actually looking at the species composition of the diatom or dinoflagellates, really all the plankton. So uh, I don't know if they use artificial intelligence to interpret their images, but they have a system which can look at the microalgae and determine if red tide is present or not. It's a very
Mm,
difficult system to keep running. Um, Depending on how you define it, a lot of the stuff that we work on as an agency is restricted because we are technically a law enforcement agency. So, for example, if I wanted to put a camera on an oyster reef and see if people were poaching, I can't do that because that's technically an invasion of privacy. So I have to be really careful, for example, about the use of drones. If I wanted to put a camera on a boat ramp, um, if there's any possibility that, for example, it might record license plates that are using that boat ramp, I can't do that. And
mhm,
so it's the structure of our agency because of that law enforcement component. It's tricky to use things like um, remote sensing that measures human behavior. I'm not 100% sure. For things like I put up the picture of the diseased animals, we are trying to develop software where we can analyze our samples faster. In
Mhm.
other words, if a female oyster is pregnant, how many eggs are there present in her gonad? Um, and we have used those in the past. We're trying to not necessarily use artificial intelligence yet. There are softwares that do things like image analysis, which essentially are kind of artificial intelligence. They're really more just simple algorithms. So, in a sense, but not, um, I wouldn't say virtual reality. Um,
Yeah.
yeah.
Right. All right. Two more questions. Where do you believe the most impact can be had by citizen scientists? What kind of data collection by them, if any, has been beneficial for your studies?
Yeah, I mean, so there are things that I have participated in. Historically, I used to do a thing called the scallop searches. We still have one in Tampa Bay called the Great Bay Scallop Search. We did some of those in Pine Island Sound. Or I shouldn't say we. SCCF in conjunction with the University of Florida extension agents. Um, the Charlotte Harbor National Estuary Program used to do something, and I don't know if they still do it, where, for example, you have, I mean, coastal cleanups are great, but they would do things like, you organize a team where they go out, for example, and one of the efforts is they did as much of the Charlotte Harbor estuary as possible to assess whether the shorelines had been hardened or not. So is
Mhm.
it a natural shoreline or is it developed in some way with a seawall or riprap? So there's a lot of opportunities like that. I know the museum has a thing where you go out and you enlist volunteers to go, what are the common species still common? What is present on
Mhm.
the beach?
Mhm.
So there's lots of programs like that. I know SCCF does that. We have a group here called Tampa Bay Watch, where they're, for example, planting plants, uh, building oyster reefs, etc. So there's a lot of opportunities if you search for them. Sometimes
Mhm.
you might not be able to do the scale of restoration that we can do, when we
Mhm.
get these $1 million grants,
Mhm.
but they're absolutely helpful. But if you go to things like iNaturalist, there's a
Yeah.
little bit of question sometimes with the data quality, but I think it's not necessarily, pleased isn't the right word, but there's enough feedback where if it's really erroneous, it might get monitored. But
Mhm.
iNaturalist is a program where people can report sightings. I saw this thing in this place.
Yeah.
And we certainly use those, not necessarily for all of our science, but sometimes as starting places.
Yeah. Right.
So there's a lot of different kinds of those opportunities. Um, and then from my own personal experience, um, especially among retired people, I have experienced an attitude where, um, I don't think elitism is going too far, where some scientists undervalue the value of professionals. And
Mhm.
some of the retired professionals I have dealt with, for example, one of the guys I dealt with designed the toilets on the space shuttle.
Mhm.
And so,
Right.
yeah, he's not necessarily a professional oyster biologist.
Right. It's pretty good.
He was a pretty smart guy. And so, and I know anybody that is familiar with the shell collecting world, a lot of the shell collectors were not trained malacologists. And a
Yeah.
lot of them are way better than I am at malacology.
Mhm.
That's one that is near and dear to me. Um, and so you may not be an academically trained expert, but there are a lot of amateur quote unquote experts that really, really are passionate about what they do.
Sure.
Another one is bird watching. A lot of bird
Mhm.
watchers are not trained ornithologists, but man, they're good. So there's just a ton of ways that volunteers, quote unquote, can contribute to science. Um,
Yeah.
and I have a lot of respect for so many people that I've met in my career. So
That's good. That's
yeah.
good. All right. Linda has a question. It's kind of a big question, I think. Are oysters and other U.S. locations experiencing similar problems as Florida?
So I mean that's actually not that hard. Oysters as
Yes.
as as a species or as a multiple species around the world have had problems. Oysters generally rely on estuaries. Human development generally is centered around estuaries. And so there's a great book called The Big Oyster. If you ever really want to read the history of
Yeah.
oysters, phenomenal book. For many, I mean, for centuries, oysters were viewed as something that which you could never overharvest. They were just, boundless. I'm trying to think of the right word. Um, Clearly that was not the case.
Limitless, right?
Limitless,
Yeah.
thank you. Yeah.
Yeah.
There's a lot of reasons why oysters in general, writ large, are threatened. Oysters as an aquaculture species have done much better than natural populations, but they are hard to grow. They're not necessarily an easy species to culture. There's a huge pearl industry, pearl oyster industry, um, whole different conversation, but has been very successful. So in general, yeah, oysters in many places are threatened or endangered. Another thing that has happened with oysters is because some species are easier to culture than others, not every country in the world has the same ethos or uh, objectives or needs as maybe the environmental community in the US.
Hmm.
There are many places in the world where they need protein, and oysters are a good source of protein.
Hmm.
So they don't care if the oyster came from a neighboring country or around the world. If that's a species that is easy to culture, their priority
Hmm.
is food production.
Hmm.
Um, what that leads to is, a lot of species have gotten moved around the world. And I don't want to judge that because that is, like I said, the interests in other places, their priorities may be different than ours.
Sure. Yes. Yeah.
And so an, another aspect of that is that, some species are getting moved around the world. And so maybe
Hmm.
biodiversity is improved. I don't know. I'm just trying to
Hmm.
think of a nice way to say that.
Hmm.
But for the most part, one of the things that happens when you move aquaculture species around is that you bring disease or predators. So green crab is an example. Green crab was introduced to the U.S. East Coast, and then some of the tunicates, which are a detrimental predator, and the tunicates, sea squirts, can smother benthic habitat. So
Hmm.
there's many aspects to moving species around. In general, oysters have suffered, and pretty much all species of oysters, almost anywhere.
Right.
Yeah,
It's
I
the way
mean,
of the world.
unfortunately, that is the way it is.
Yeah, way of the world. Right. All right. Last question from Jerry, who says, excellent talk. Uh,
Thanks, sir.
Jerry's question regarding the four study groups, is there a control model or group used for comparative analysis?
Yeah, so when we first designed this, and I didn't put that slide up, we had what are called outgroups, and so our study stations actually included Biscayne Bay, Lake Worth Lagoon, which we do still study, Sebastian River, Mosquito Lagoon, and Tampa Bay. The idea when we first established that, and this is called uh, most good research has an experimental condition and a control condition. So we designed this study initially with quote unquote control populations that would not be affected by Everglades restoration.
Mm.
Unfortunately, Tampa Bay, Sebastian River, and the Mosquito Lagoon, um, they are all affected by management, human development, climate as well. And
Mm.
so the Water Management District, after several years, I think 3 to 6, depending on the place, recognized that there really is no true control for something like an oyster in a state like Florida.
Mm.
Um, Unfortunately, that's just that every place in Florida is being affected both by climate, by development, by restoration projects, by water management. There's a lot of water management restoration projects going on pretty much anywhere. A lot of things called mitigation. So if you cause harm here, you try to repair it somewhere else.
Mhm.
So unfortunately, we don't have in my opinion, great controls for our
Mhm.
experimental sites, just
Right,
because now
right.
we do still study
It's
other
fluid.
places.
Mm.
And we study oysters basically everywhere in the state except for the Jacksonville area right now. And there are some other groups that do that. And so we're going to have to do a different kind of statistical analysis, which is a little bit harder, because we can't have true controls. And it's just, it's a great question and something we struggle with. It makes your statistics to really show that there was or was not change a lot harder. And
Right.
that's part of why I try to caveat when I talk about is these are patterns that we see. Trying to attribute cause to those patterns is a great challenge in most cases. Yeah,
Yeah.
it's hard.
All right. Well, Mr. Geiger, thank you very much for
You're welcome.
sharing your expertise and your studies and your perspective and for being part of this talk series. Thank you to all attendees who joined, and thank you for your questions. And everyone go forth and and keep paying attention and doing what you can to make things a little bit better. And
I
thanks,
appreciate
Steve,
it.
thanks for giving us some more information to do that.
I
So
appreciate the chance, and thanks to all the people who managed to listen to this entire thing through the whole thing. I know I talk a long time, so I appreciate it.
yeah, you do it well. You do it very well.
Thank you.
Thank you, and it's good to see you. And
Have a great evening.
okay, right. Thank you.